Oh, exciting. Now we can finally work with the autonomic nervous system part 2. As we looked in part 1, we saw that how the system worked. Now we can use it in an actual real life example and show how we could move through this and see it in this turtle. Anyhow, let's begin. So the first thing we need to understand is how these neurotransmitters work. The main focus and most important point here is the second one, that the physical act of binding causes this action potential. So to start, there's a unique form of a neurotransmitter, and what we see is that the neurotransmitter uniquely fits with and binds to a receptor on a neuron. And it is, again, this physical act of binding that causes the action potential. Then what we see is that once that action potential has happened, the action potential is going to travel down the axon and change something in the neuron or signal something else further down in the neuron.
Well, I'm wondering, what are you doing?
Oh, yeah, right. Location of these is that we find them on the cell bodies of postganglionic neurons, so a second neuron in series, so after the ganglion. And then we also find this same type of receptor, the nicotinic receptor, in the adrenal medulla, and then also within skeletal muscle. Conversely, we see that the muscarinic receptor is found on smooth muscle, cardiac muscle, and glands. So, for these ACH receptors, what do we expect to bind? Well, for a muscarinic receptor, muscarine will bind, as well as ACH. And then for a nicotinic receptor, nicotine will bind, and we also see that ACH will bind. Now, is it surprising that ACH can bind to both of these and that these two different chemicals are binding to the other types of receptors? That is, if we look at a muscarinic receptor, what binds? Muscarine, but not nicotine. So nicotine cannot bind to a muscarinic receptor, and then conversely we see that muscarine will not bind to a nicotinic receptor. So we see muscarine being able to bind, but not nicotine, and then ACH. And then for the nicotinic receptor, we see nicotine can bind ACH, but not muscarine. So is that surprising in any way? In other words, is it surprising that ACH can bind to both of these types of receptors? The answer should be absolutely not. Why? Because these are ACH receptors. So of course you're going to see ACH binding to them. Another type of receptor, norepinephrine receptor. Two types here, alpha and beta. For alpha and beta there can be subgroups. So let me explain that. If we look at the subgroups, what we would see is that we might find things like alpha 1 or alpha 2 or alpha 3. And so these subgroups, different types, are going to have some interesting challenges for us, and we'll explore that later. Similarly, what we see within beta is we could have beta 1 and beta 2 type receptors. Again, these are Greek letters, so that's why the funny shape of them. So the alpha receptors will be found on smooth muscle and glands, and the beta receptors will be found on smooth muscle, glands, and cardiac muscle. So just again, for a difference between the norepinephrine receptors and the nicotinic and muscarinic receptors, the ACH receptors. So, to get into this experiment, what branch of the autonomic nervous system does the vagus nerve belong to? Well, from what we've covered so far in this course, you should have a pretty good idea of what that answer would be. Okay, so the vagus nerve, does that ring any bells? It should. It's cranial nerve number 10. And when we were looking at cranial nerves are part of the parasympathetic nervous system. So we should know that the autonomic nervous system and the vagus nervous system are parts of the parasympathetic nervous system. So parasympathetic is rest and digest, and so we should see that if we're looking at this experiment in terms of a heart rate for a turtle, rest and digest, you should see that the heart rate would be slowing down.
Okay, let's look at the experimental objectives then. The first experimental objective we want to deal with is to determine the effect of vagal stimulation on the heart rate of the turtle. The second one is that we want to determine the effect of nicotine on vagal stimulation. And the third is that we want to determine the effect of muscarine on vagal stimulation. These experimental objectives do not say that we are dealing with the effect of nicotine on the heart rate of the turtle. So notice that the first one is the effect of vagal stimulation on the heart rate, and the second one is the effect of nicotine on vagal stimulation, not the effect of nicotine on heart rate, which is important, and we should think about that and circle back around and make sure we do get that. Okay, so let's start by looking at the effects of vagal stimulation on the heart rate of the turtle. What would we need to do? Well, what we would need to do is isolate the heart of the turtle and isolate the vagal nerve. And so through some cut downs of removing the plastron, that's the bottom shell, and exposing the carotid artery in the neck, we would find that attached to the carotid artery is the vagal nerve. And we could attach a stimulator to that. And then we could attach a force transducer to the heart and then look at how much it's moving. And so that you could record the heart rate of the turtle and you could record whether or not we're putting stimulation into the vagus nerve. Okay, so let's see what this would look like. Now, as we would do this, we want to determine the effect of vagal stimulation on the heart rate of the turtle. So how would you determine that? What would you look at? Well, the first thing that you would need to do is have a control. So the very first thing that we're going to do is just record a normal heart rate. Again, our computer printout would have the heart rate at the top and whether or not we're vagally stimulating on the bottom. So a solid line on the bottom is going to be no stimulation, and then some sort of hash marks on that line means that you are pushing the button and stimulating the vagus nerve. Okay, let's continue. So this would represent our normal heart rate. So again, we're going to record heart rate at the top, and we're going to record vagal stimulation at the bottom. And here, this nice sine wave is the contraction, relax, contract, relax. Eject the blood, fill, eject, fill. And that's what's happening with the heart of the turtle. And again, this one is just a flat line because if we had some sort of markings on this, it would say that we were stimulating. But for normal, for the control, we do not want any stimulation on there. Okay, so let's go to the first objective, and the first objective is to determine the effect of vagal stimulation on the heart rate of the turtle. So if we want to do that, what do? Well, we push the button. So here we see the stimulus. Okay, so here's the heart rate cruising along normal. When the stimulus starts, the heart stops. And then it gets this extra large beat, and then it resumes completely normal after that one large beat. Okay, so that's what happens when we stimulate the vagus nerve. Now let's go on to the second part, and that is to determine the effect of nicotine on vagal stimulation. Excuse me. So if we are looking at adding nicotine to the prep, so adding the nicotine around the heart, what we will see is that the heart rate remains completely normal even during stimulation. So recording the heart rate, recording stimulation, I see we have stimulation, but it does not affect the heart rate at all. The heart rate remains exactly the same. So at this point, we don't exactly know what's going on, but what I do know is that when I'm putting on the nicotine and then I'm trying to stimulate the vagus nerve, there's some sort of blockage that goes on. But if I think about when I'm pushing that button, what am I actually trying to do? Well, I'm trying to release acetylcholine. So let's go ahead and add some acetylcholine and forget to try to release the acetylcholine this way, but release the acetylcholine just by dumping it on. So if we just dump on the acetylcholine, what we see is all of a sudden the heart takes a very different beat, and all of a sudden we see that it's these huge beats, big pause, huge beat, big pause, huge beat, big pause. Again, nothing on the stimulation line because we've switched to try to bypass this system and looking at how it works by just putting on the acetylcholine. So when we put on the acetylcholine, we see these very large beats and these big pauses over and over and over again. Okay. The next thing that we'll want to do is we will want to move on to the third objective, and the third objective is determine the effect of muscarine on vagal stimulation. So let's look at that next. Now, before we look at this muscarine, what I want to do is I want to make sure that the whole system is operating normally again. And so in order to do that, what I would want to do is I'd want to be able to repeat the normal, and then I'd also want to be able to repeat the vagal stimulation. So I want to repeat number 1 and number 2 again before I go on to this one, which is number 5. So we get everything flushed out. We're back. We repeated number 1. We repeated number 2, the vagal stimulation. And now we dump muscarine on in the same places that we put the nicotine, and we are moving along just normally again. Even though we hit stimulation, we still remain normal completely through that. So we say, okay, well, last time we added some acetylcholine, and that worked very differently. So let's look at that. And here we see if we add the acetylcholine, we remain completely 100% normal. And again, no stimulation here, but we stay completely normal, and there's not the big change like we saw the last time. Okay, well, now let's go through an analysis of what's happening with each one of these. The best way to figure out exactly what's happening in this experiment is to break it out and look at it in terms of a schematic. A schematic is just a graphic representation of what we're looking at. And the reason I say that it's a representation is because we're dealing with the parasympathetic nervous system, which means that our autonomic ganglia is a terminal ganglia, and it's located at or within the effector organ. So this slide doesn't show anything like that. Here's our terminal ganglia way over here. Well, we correct that by putting this little dashed line or this little solid line here and saying that everything over on this side is behind the heart. Good. So now we fixed the schematic so that it works a lot more like, and it is spread out so we can tell a lot of what's going on. So we need some players in this. And first of all, we have nicotinic receptors, and those are located on the cell bodies of the postganglionic neurons. And so those are shaped as little backward C's. And then we have muscarinic receptors, and those are located on the effector organ. And then what I need is I need these chemicals to be able to follow the truth, and that is ACH must be able to bind to nicotinic receptors. So this end plugs right into the C section, and this point would go right into the points of the muscarinic receptors. So acetylcholine can bind to nicotinic receptors and muscarinic receptors. Nicotine can block right in here and do nothing over here, and then muscarine can't do anything over here, but certainly can block in this region. So there's how all of these players are going to work out. The difference, though, we want to make sure we know the difference, and that is when acetylcholine binds here, you get an action potential. When acetylcholine binds here, you get an action potential. So action potentials are going to be very different than when the blocker is in place. The nicotine, if it binds here, it just takes that spot. It doesn't actually have an action potential that's happening. So we would block the acetylcholine from coming in here. Same thing with muscarine down here. When it's in there, it doesn't send an action potential, but it does occupy the space and not allow for acetylcholine to go in there. Okay, so let's look at this and find out what would happen if we just push the button. So we're going to put a little button in over here, a little zap over here, and then follow through what's going to happen. So we zap this right here. That would trigger an action potential. That action potential would travel down to the terminal of the preganglionic neuron. Out of the preganglionic neuron, we would expect acetylcholine to come out. So acetylcholine comes out, crosses the synapse, binds to the nicotinic receptors, triggers an action potential within the postganglionic neuron, and then that travels down to the end down here, which causes the postganglionic neuron to release acetylcholine. The acetylcholine crosses and binds at the effector organ. When we get acetylcholine binding at the effector organ, we slow down the heart. So what does that look like in terms of our slides? We see that before we're pushing the button, we get the normal heart rate. While we're pushing the button, we completely slow down the heart. In fact, we stop the heart. And then we go to a very large heartbeat afterwards. Now this is interesting because we did slow down the heart, and that does explain what's going on there. But now let's talk about this. All right, this is referred to as Starling's law of the heart. And Starling's law of the heart states that if you increase the preload on the heart, you will increase the force of contraction. In fact, that works for Starling's law in general on any muscle. If you increase the preload, you increase the output. Okay, so what does it mean to increase the preload? Well, let's look over here. If we look over here, what we see is you are contracting, so you eject the blood, fill, eject, fill. Now what happens? Fill, 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 fill. So all this extra filling says that I've increased the amount of preload, I've increased the amount of blood that is in the heart. So now the next beat is going to be very forceful to eject out that extra blood that is stretching the heart. Then, so I eject rapidly or vigorously, and then I fill, and then empty and fill. So I'm right back to normal down here. So this extra large beat is because of all this extra filling time. So increased filling time, increased force of contraction, increased preload, increased output. Okay, so that's Starling's law. All right, so we definitely see that we are able to slow down the heart. Now let's move on to another part. We'll add the nicotine. So here we dump on the nicotine, and the nicotine goes everywhere behind the heart. We get all the nicotine over here. It's not doing anything. It's just in that area. But here we see it binding, and it's binding to our nicotinic receptors on our postganglionic cell bodies. So now what would happen if we push the button and zapped over here? We zap, action potential comes down. Does acetylcholine come out? Yes. Acetylcholine comes out, and acetylcholine can't do anything. So then it lasts for a little while, a couple seconds or a second or two, and then it's gone. So can we get anywhere else past this? No. So nothing else is going on here, and so no change in heart rate. So let's look at that in terms of one of those diagrams again. And as you can see, we're moving along normally, and the acetylcholine, the acetylcholine is trying to be released by stimulation, but we're not getting there. We just don't get there. If we can't get to the heart, we can't change the heart rate. So then we move on and we want to add acetylcholine. So we dump acetylcholine everywhere behind the heart. So the acetylcholine is going over here, not changing anything there. But if acetylcholine is here, it can interact with the muscarinic receptors to slow down the heart. And that looks like this. So we get the big pauses, big beat, big pause, big beat. Again, this large beat is Starling's law of the heart. And that's, again, in all of these repeated because Starling's law, fill, 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 fill. Starling's law, fill, 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 fill. Starling's law. So we get the increased filling time gives us that huge beat. And so it takes on that very characteristic look because of that. Okay, so that's looking at what's happening on the nicotinic side. Now let's go to the muscarinic side. Now, before we do that, what do we got to do? Well, we have to wash this out. So here it says, is it washed for real? How do I know? And the only way to know would be to go ahead and push the button. So pushing the button triggers an action potential, gets us to there. It moves down here and jumps across that synapse, and we slow down the heart. So we get the opening here, and we go to a repeat of the number 2. So we'll get back to normal beating, but how do I make sure it's washed? I'm able to repeat the vagal stimulation portion. Okay, so now if we go to the third objective, which looks at the effect of muscarine on vagal stimulation. So muscarine on vagal stimulation, I dump my muscarine in, it doesn't do anything here. I dump my muscarine in and it binds here. Okay? Now, when I stimulate here, I get my action potential to come down, it goes across, it binds, goes across the synapse, binds here, triggers an action potential here, action potential travels down, causes the release of acetylcholine here. So the acetylcholine is being released here, and we're bound, so we can't change the heart rate. So what does that look like? Looks just like this, where we have the normal heartbeat, and even during stimulation, we don't change that. Okay, now let's go to the one where we add the acetylcholine. Now, if we're adding the acetylcholine and dumping it on all back here, we get acetylcholine binding to these nicotinic receptors. That might even trigger more acetylcholine coming out here, but all the acetylcholine down here isn't doing anything because we are blocked. So can we change the heart rate? And the answer is no, there's no change in heart rate. So cruising along like this with the addition of ACH, no stimulation, and we get no change there. So that's pretty much what happens with the system. And what we're seeing is a very clear representation that the schematic is telling us everything. So let's go to another picture of the schematic and look at it all together and make sure we do understand it. Okay, so here we see that this system is the 2 neurons in series. Nicotinic receptors up here, muscarinic receptors down here. So the key to understanding this is looking at this in 2 different ways. So you have the times when you are stimulating here and trying to get all the way through the system. I call that moving from point A to point B. Can we get there using the system? And so that's any time that you are pushing the button, any time that you are sending an action potential through the system. If you're blocked here, can you change the heart rate? No. If you're blocked here, can you change the heart rate? No. So there's no effect if you're blocked here or if you're blocked there. The second way to look at this system is, what if you are adding on the acetylcholine? So you're not pushing the button, you're adding on the acetylcholine. So what matters? Well, we saw that in the second time with the muscarine, if you don't have anything blocking here, you could get the neurotransmitter binding there. But it doesn't matter. The only thing that matters when you're adding the ACH is, are these receptors open or not? So for the exam, what you will see is, maybe, for instance, I say, well, everywhere there was a nicotinic receptor is now a muscarinic receptor, and everywhere there was a muscarinic receptor is now a nicotinic receptor. So we switch these. Could you follow through the exact same experiment again using that? And the answer is, I hope so. Okay, so let's go through. We understand the schematic. The key to doing well on the exam is recreate the schematic. Once you've recreated the schematic, the answers will come to you very naturally. The one thing we didn't answer is, what's the effect of nicotine on just the heart rate? And why does your heart rate increase when you're a smoker? Or why do you have increased heart rate with somebody who would have nicotine in their body? That goes back to the idea of the parasympathetic nervous system always being on. If the parasympathetic is always having an output, and we put a little bit of nicotine in here, we're going to block these. every once in a while. So you're going to block the parasympathetic. So what is the parasympathetic doing? The parasympathetic is putting a parasympathetic tone on the heart. So it's slowing down the heart a little bit all the time naturally. So with the nicotine in there, the nicotine is blocking every once in a while, so your signals aren't getting through to slow down the heart, so your heart rate would be higher if you're a smoker and you're bringing in nicotine. All right, let's go to the experiment. So this you should know. These are the things that you need to know. You're going to record normal. You're going to electrically stimulate the nerve. Then you add a blocker and electrically stimulate. Then you add the neurotransmitter. Then you wash the whole thing and make sure that it works. Then you go to adding a different blocker and electrically stimulating, and then you add the neurotransmitter. So 3 and 4 is the same as 5 and 6. In the experiment we just did, we recorded normal. We electrically stimulate the vagus nerve. We add a blocker, nicotine, and then we try to electrically stimulate. And then we add the neurotransmitter acetylcholine. Then we wash the whole thing out. Then we add muscarine and electrically stimulate. And then finally we add the neurotransmitter acetylcholine again. All right, so 2, 3, and 5, that's when you electrically stimulate. And 4 and 6, that's when you apply the chemical stimulus. That's when you apply the neurotransmitter. All right, so just as a total recap, here is the entire set of recordings. I included them all in individual slides, but here they're all together so you can see exactly what's going on in our experiment. Remember, for the exam, I could use sympathetic, I could use parasympathetic. I could, again, the example I gave in class is I could replace nicotinic receptors with muscarinic receptors, and then muscarinic receptors with nicotinic receptors. So basically changing the location of those. But there's many different ways that I could ask these questions on the exam. The most important thing for you is to understand how the system works and using the schematic, following through with using the schematic and the steps of the experiment that you know and you commit to memory. Once you do that, I think the answers for the exam come pretty regularly without too much trouble, and most people can follow this through and get it. If you have any questions about this, please ask them.